So tonight, I'm going to be talking about the tour of a tiny city, an excursion into a biological cell. And before we do go to the next slide, I'm going to give you the conclusion of the talk, which is that the cell is very complicated, more than anyone ever thought possible, and that we are only going to touch on a number of things in the cell, but there's even more than I will present in this talk. So cells are the building blocks of organisms. The single-celled organisms, the yeast, the amoeba, and some algae shown here are just made of single cells, but then there's the multicelled organisms, that's plants and animals, and just about everything you can see are multicelled organisms. And there's some multicelled organisms that you're not even able to see, that they're too small. The third group are slime modes, and these are really interesting organisms. They live as single-celled organisms, except when they go into starvation mode. And once they're starving, they are able to become multicelled organisms with the ability to move, build reproductive organs, and reproduce and produce new slime molds. And then once they have food around again, they go back into being single-celled organisms. So the discovery of the cells was first done by Robert Hooke in 1665. He was an English scientist, and he coined the term cell because what he saw using a primitive microscope reminded him of the small cells in which the monks lived. They lived in monasteries, and each of them had their own little room. And when he saw the cells that under the microscope, that reminded him of the cells that the monks lived in. And then in 1833, which was almost 200 years later, the microscopes got good enough that Robert Brown, Scottish botanist, could see the nucleus. And then Rudolf Wagner, a German biologist, described the nucleolus, and that's this dark circle inside of the nucleus right here. So this is a cartoon of what Darwin knew. So he knew about the cell. He knew there was a nucleus. He could see the nucleolus, but he thought that it was a simple bag of protoplasm, and that life was just simple. And so his idea of life starting in a warm little pond, like he called it in his book, he thought that was very possible because it was a very simple thing that the cell was made of. Most of the information that we know now, and here's a picture of a diagram of what the cell, at least partially what the cell looks like nowadays. Most of this information was discovered within the past 50 or 60 years. So there was not a lot known when Darwin developed his theory of evolution. So we're going to be looking at the cell here. And if you think of the cell as a city, what does a city need? If you think about where you're living, what are kind of some of the services that are provided by the city? And if this was a live audience, that I would ask you to participate, but we're just think about what kinds of services you could see. What I thought about was the city hall, the library, moving companies, manufacturing plants, post office, waste treatment plant, power plant, storage facilities, streets and roads, and city gates. And in addition to that, this city, this tiny city, has the ability to reproduce, move, and build more manufacturing plants, power plants, streets, and roads. So we're going to start with the city hall. In other words, the nucleus. We will get to the blue dot in the center here is the nucleolus, and we will get to that later. We'll look at the nuclear pores next. Here's a diagram of the nuclear pores. Looks like a hole. These little tiny holes here that are in the walls look like they're just holes, but they're not. They're a very complicated pore which provide a guarded gate of what goes into and out of the nucleus. There's a lab that I worked in, they studied one of the proteins that makes up this nuclear pore. And when that one protein did not work, the whole cell would shut down and it could no longer divide just because the one protein that made up part of this nuclear pore was not functional. And so each one of these proteins that are in this pore are very important. Not only do they guard what's going in and out of the nucleus, they do it in a very rapid way. This nuclear pore can handle about 1,000 molecules per second. And then most of what's inside the nucleus is DNA, which stores almost all the genetic information. The DNA is made into RNA, and we will look at this, how this RNA is made from the DNA in this next slide. So the nucleus you can think of as also a library. It stores information. Every cell has the same genes, but if you think about it, how does a liver cell who has all the same genes know which genes to transcribe for a liver and not transcribe for the genes needed for a muscle cell, for example? How do they know how to do that? Well, if you're in the library, you would ask a librarian. So librarians find the information. So in this diagram, the gene is downstream here, and we're looking at the upstream portion here. And the librarian could be thought of, the transcription factors could be thought as librarians. And what this is that if you are needing to transcribe a particular gene, for instance, if the cells go into heat shock, that the temperature goes too high for the cells, there's a whole heat shock response in the cells, and they send out the transcription factors, which will bind to the transcription factor binding site here. And we'll look at that in the next slide here. So here's a picture of the DNA. The gene is downstream here, and here is a certain 6 to 8 sequence letters that are the transcription factor binding site. And if the gene downstream is going to be transcribed, the first thing that happens is that a transcription factor will bind to the transcription factor binding site. And then after that is bound, that will recruit about 6 to 8 more proteins. And that is known as the transcription polymerase. And that will go downstream and transcribe the DNA into the mRNA. Once the DNA is transcribed into mRNA, there's a couple steps before it can do that. Here, if you see that DNA, the gene, is not contiguous, there would be a piece of the DNA here, and then a piece of the DNA here in the exon, and then here in the exon. And in between, there are pieces of DNA that are not instrumental in getting the all the contiguous DNA. And so what happens is that there is an organelle called the spliceosome. And what this organelle does is that it will take the initial transcript here, cut at this point, cut at this point, and put the two pieces together and bind it here. And so in the end, what you get on the end of all this going through the spliceosome is an mRNA that is ready to be translated into a protein. And then it gets sent out through the nuclear pore. This is a picture of an mRNA. M stands for messenger RNA. There are about 5, 6 different kinds of RNA. We're going to be just talking about 3 types. This one is an mRNA, which stands for a messenger RNA. And this is a picture of one of the mRNAs exiting the nucleus through the nuclear pore. At the end of this talk, I'm going to show you a short 3-minute movie talking about the the different organelles, and some of these pictures I've taken out of the movie, and this is one picture to watch for. It goes very, very quickly though, so just keep this picture in mind. So once you go the transcription into mRNA, it goes out of the nucleus into the cytoplasm, and it gets translated into proteins. Well, for that, you need manufacturing plants, and that's done by the ribosomes. I think that the ribosomes are one of the two most complex organelles in the cell. And it is, if you look here, all these little dots are the ribosomes here. And you can see them, they're just covering all the way through the cell. All these dots are ribosomes. So we'll look at that and how that is done. Here's another picture on the left that is a picture that I pulled out of the movie you'll be seeing. This is the mRNA coming down through here. The ribosome is reading it. There's a small subunit, a large subunit, and here's a piece of protein that is being manufactured as it reads this piece of mRNA. And on this cartoon here, we'll look more at the inside of what goes on in that ribosome. So again, there's a large subunit, there's a small subunit, and there's 3 slots, 1, 2, 3. And the mRNA is coming through here, and the ribosome is reading it. These little cartoon pieces here that look like chess set, the pawns in a chess set, are really tRNA. This is another type of RNA, and the T stands for transfer RNA, because what it can do, what it does is that it has the signal for what amino acid needs to be attached up here. But the reading, what the ribosome reads, is this piece down here of the tRNA. And so if you look over here, this would be the tRNA, which really looks like the picture on the right more than that. And it has an amino acid, and it comes down into this first piece here. And if these 3 bases correspond to these 3 bases of the mRNA, then it will attach. If it doesn't, it gets kicked out and the next one comes in. And again, there is a lot of reading of making sure that things are done correctly. If they bond here, then it moves on to the next slot here, and the protein that was attached to the tRNA will then bond to the growing amino acid chain right up here. And then it moves over to the next slot and gets kicked out. And again, there's a lot of proofreading, and also it does this proofreading and the building of the protein chain very quickly, and it can do this about 20 amino acids per second. So how are the ribosomes made? Well, ribosomes are made up of ribosomal proteins and rRNA. Now, this is a third type of RNA. There's the messenger RNA, the mRNA, and then there's the transfer RNA, and now this is ribosomal RNA. And in the nucleus, there's DNA that codes for the messenger RNA, including the messenger RNA that codes for the proteins in ribosomes. And DNA also codes for the transfer RNAs. In the nucleolus, the DNA codes for the ribosomal RNAs. And what I find really incredible is that the ribosomal proteins are made by the ribosomes. So they need to have the ribosomes to make the proteins that make up the ribosomes. How can this occur by chance? How would that have happened? Something to think about. So what happens when proteins are made? How does it know where to go? Well, the post office, you'd have to have the post office tag the proteins, and that is done by the endoplasmic reticulum and the Golgi apparatus. The endoplasmic reticulum are responsible for the transport and some metabolism and production of some steroids. There's the smooth ER. Here's the rough ER. The only difference between the smooth and the rough ER is that the rough are rough because all the ribosomes are attached closely to this, to the endoplasmic reticulum. And so they need to be packaged and labeled and sent out more quickly than the smooth endoplasmic reticulum. And then the third one is the Golgi apparatus, which works with a lot, setting up proteins and sending them outside to be secreted from the cell. So here is a picture of the endoplasmic reticulum and the Golgi apparatus. The dots, again, are the ribosomes, and the endoplasmic reticulum folds and tags the proteins for their destination. The Golgi apparatus encloses those proteins, which are just destined for secretion, and then the as you see, the little vesicles, secretory vesicles, will go to the plasma membrane and then get secreted from the cell. For example, if this is a white blood cell that is manufacturing antibodies, the antibodies have to get out of the white blood cell, and so they will be secreted through the membrane. So where are we so far? We have the city hall and the library. We've looked at manufacturing plants and post offices. Next thing we're going to look at is the streets and roads and the moving company and some storage facilities. So these are microtubules. There's mainly two types of tubulin that make up microtubules. There's the alpha tubulin and the beta tubulin, and they fit together to form these long tubes. And they'll be precisely, I think it's 11 tubulin molecules per to make up the diameter of these microtubules. And they will polymerize. Here's a picture of the microtubules polymerizing at 100 monopers per second. And then they depolymerize at 1,000 monopers per second. The microtubules have a lot of different jobs in the cell. One of them is structure. It provides the structure of the cell, sort of like 2x4s in a building. And we'll see pictures of all three of these in the movie that I will be showing you at the end of this talk. The other thing microtubules provide are streets and roads. And here's a picture of a kinesin, a motor protein, carrying a vacuole on a microtubule. So if you look, here are the 2 feet of the kinesin, and this is called a motor protein. So this will actually walk, put 1 foot in front of the next and walk along the microtubule, carrying this vacuole that is going to be sent out to the outside the cell wall. And we'll see this in the movie. Here's another one in the background here carrying another vesicle. And these vesicles, you can think of them as storage facilities because they've been packaged up with all the proteins that needed to be sent out, and they get packaged and carried over to where they will be sent out to the cell. There's also, the microtubules also act as moving companies. They push and pull things around. And this picture, if you look, the blue in the center here are the chromosomes. They're all lined up. This particular cell is about getting ready to divide into 2 new cells. So there's duplicated chromosomes of every kind of chromosome. There's 4 of them instead of 2 of each kind. And so then these are the microtubules. The green things are the microtubules. This is what's forming the microtubules. They call the microtubule organizing center. And the microtubules will get sent out, grab each chromosome, and then it depolymerizes at this point. And so the microtubules get shorter. And as it gets shorter, it pulls the chromosomes apart. And then the new cell wall will come down through here, and then you have 2 new cells, each with a proper number of chromosomes. So those are the microtubules. So we have 3 things to go, or a couple of things to go yet: the waste treatment plant, the power plants, and the city gates. So we'll look at those. The waste treatment plant could be, the lysosomes could be thought of as the the waste treatment plant. And here are the lysosomes. Here's the little blue things here. And we're not covering like the peroxisomes or the centrioles or those things. But here's the lysosomes right here and here. And their job is to break down excess or worn out cell parts. They may be used to destroy invading viruses and bacteria. They have a very high acidic enzymes inside the lysosome, so they can digest anything that they want to try to get rid of. And if the cell is damaged beyond repair, the lysosomes can help it to self-destruct in a process called programmed cell death, or apoptosis. Sometimes in early cancers, the cells divide. The problem with cancer is they divide very quickly without having any of the proofreading of what is going on in the cell. And that's what the problem is with cancer, is they've lost their controls. And in early cancers, a lot of times the lysosomes will self-destruct the cancer cells and get rid of them. But the cancer cells divide so quickly, a lot of times it just gets away from it. And so then you have this cancerous growth in organ, in some system. The next thing we'll look at is the power plant. And this probably is the next most, or the other most complex organelle in the cell besides the ribosomes. So we'll look at the mitochondria. And they're the ones that produce the energy that we can use in our systems, or all animals can use. So where do we get our energy from? Well, all energy comes from the sun in one way or another. And the photosynthesis, plants do photosynthesis. So they take the sunlight, energy from the sunlight, and they take water from the ground and carbon dioxide from the plant, the air, and put that together with the energy that they get from the sunlight, and they manufacture glucose and oxygen. And this process is called photosynthesis. Then we eat the plants, or we eat the animals, the meat from animals that eat the plants. And we have the digestion gets broken down from the glucose, and that together with oxygen, it produces energy and excretes water and carbon dioxide. So if you look at this, it's a complete system. It's the water, carbon dioxide, energy from the sun gives glucose and oxygen, and we take the glucose and oxygen, produce energy in the form that our bodies can use, and produce water and carbon dioxide, which is the beginning parts of this. So it's pretty much a transfer from the energy from the sun to the energy that we can use. So this is the energy production, and it's done within the mitochondria. And it's a very complicated process. It starts in the cytoplasm, look over here, and through a process called glycolysis. And I was at a conference earlier this year in Washington State, and there was a talk on just this one glycolytic cycle here. It was a whole hour-long talk on how all of this is done, because there's a lot of enzymes that go in and out, and control mechanisms of how to convert the sugar, the glucose, into a product that our bodies can use, and that is put into a molecule called adenosine triphosphate, ATP. Anyway, so then after it gets a couple bonds of that ATP here, then they can go into the mitochondria, and then from there it takes the leftover of the glycolysis. If you can read this, probably can't read the bottom part here, but the end product of glycolysis is pyruvate, and the beginning molecule, which is the pyruvate here, goes into this, what's called the Krebs cycle, and it goes through a lot of different chemical reactions to produce more ATP, which is a type of molecule that we can use to fuel and being able to run, to breathe, to do everything. That's how we get our energy, is using the energy within the molecule of ATP. And here is a picture of the molecules called ATP synthase. These are located in inner membranes of the mitochondria. And what happens is these center parts here rotate. And then as they rotate, it makes this protein wobble back and forth. And it takes somehow it takes the glucose, the energy out of the glucose bonds, and puts it into the ATP molecule. And you'd have to read up on it. I'm not sure exactly how all of that is done, but it's done that way. It's very complicated. And again, think back to what Darwin thought the cell looked like, and just what we know, and we still don't know everything about a cell. And what things look like that we know about now are way more complicated than what he thought of. So the last thing that we need to look at are the city gates. We've looked at everything else. So we'll look at the city gates here. So the city gates are the cell membrane. The first thing to know about the cell membrane is it's a lipid bilayer. That means that if you look, here is the outside of the cell, which is a watery substance. The inside of the cell is a watery base. The lipids, the cell membrane is made of molecules that the one side of the molecule is hydrophilic, and so it wants to be pointed either outside or inside. And the other part of the molecule here is hydrophobic. It's almost like an oil, one that doesn't like water. It's hydrophobic for that reason. The reason that this is important is that it keeps the cell wall very integral. It's not, these molecules aren't going to float away because the hydrophobic part of the molecules do not want to be by water. And so that keeps the membrane intact. And just like nuclear pores in a nucleus, there's a regulation of what comes in and what goes out of the cell. There's cross-membrane proteins that we'll look at in the next slide. And it communicates to with other cells using some of these protein signaling that we'll be looking at. So here's another cartoon of what the cell membrane would look like. And again, the hydrophilic part of the molecule, the hydrophobic part of the molecule right here. And then there are things that can go in and out, but most of the time it's very highly regulated. And then here is a picture of a cross-membrane protein. So if your body wanted to signal something inside this particular cell, but it doesn't want to go in the cell, what happens is that there'll be a protein, signaling protein, and it will bind to this part of this protein here. When the signaling protein binds to this cross-membrane protein, this part will change because of the binding. And when this part of the protein changes, then this part of the protein changes also, which sets up a signaling complex to tell the cell to do whatever it was supposed to do. So the bottom lines are that cells are highly regulated as to what comes in and goes out of the cell. So where does that leave this? Well, if the cell was this simple, it would be easy to develop in Darwin's warm little pond. But knowing, just this short little talk on what goes on inside the cell, all the molecular machines and other systems that we know now, and we still don't know everything, it would be really hard to think about how any of this could have happened by chance. And some of it is, a lot of it is so elegant, like the mitochondria and the ribosomes, just even the Golgi apparatus, that it's hard to imagine that that was anything close to being done by chance, but it was done by a designer. It points to a designer. So now I'm going to try and show this movie, and I will point out some of the things that we saw before. So I will point out some of the things. It's going to start out with inside the blood vessels, and you'll see red blood cells going through the vessels, and then they will go in, and the white blood cells will go into inside the cells, and then you'll see some of the things that I talked about in the thing. I think the music does not come through, but I'm going to narrate a little bit of but it all goes really fast. So it's only 3 minutes, so we'll see what we can do here. So there's the red blood cells, and the little blue things are the white blood cells. You're going to see some of these things. I don't even know what they are. There's the microtubules providing the structure within the cell. There's that one polymerizing, microtubules polymerizing, providing the roads. That is, like I said, it goes very fast. This is a polymerization of the cells, a depolymerization. Here comes the kinesin carrying the vacuole. And in the background, there's another one. And it really does look like that. I saw some people who made some photos of those with EM photographs. That center here is the nucleus. There's the nuclear pore, and out comes the mRNA shooting out of the nuclear pore. It will go around, and then the ribosome comes in. There's a large microtube subunit, small subunit. The protein's coming out to the left there. Sometimes they form circles, and you get a chance to see that again. This is going into a processing plant that I didn't cover. And again, there's the ribosome, protein coming out the bottom. And that's the Golgi bodies that are processing the proteins to be, the vacuoles to be sent out. And that's getting where the vesicle is sending out the proteins that were inside, or whatever it was carrying in, to be sent outside. And that's a white blood cell going to go inside a cell to look for trouble. And then you see the red blood cells in the bloodstream. And I wanted to say that this is done by, let me go back to this. Here's the references for my pictures that I had. And the movie is, if you look at the bottom line, it's Inner Life of the Cell. So you can look this up online. There's a 7-minute, the movie I showed you was a 3-minute. And then there's also the 7-minute movie of that. And then there's another page that describes more of what you see in the movie. and it's put out by Harvard University. And I'm ready for any questions, comments.
So first question is this process also regulated by signaling proteins?
Yes, a lot of them are. It just doesn't automatically happen. There's a lot of signaling. In fact, I think one of the largest signaling cascades is called a kinase cascade, Kinase. And if you look that up, it's probably 100, maybe 150 different proteins that signal in different ways for different things.
The question was, about the signaling going through the cell membrane, but also I was translating that to during the operation in the ribosome of making the transport tRNAs available and all that. Is that done by signaling proteins? How do the transport tRNAs make it to the ribosome, make themselves available? How's that regulated? I guess that's my question. Is that signaling proteins responsible for that process?
I think that a lot of that, and this is just a guess. I don't know if they even know. I don't know for sure, but I would guess that a lot of that is due to Brownian motion, because there's a lot of jiggling and going on inside the cell, and so some of that might be due to Brownian motion. But there's also a lot of signaling. There's for something to do something that is not just through diffusion, that they have to actually change the shape of a protein for something else to happen. Would be an example of when you have to signal something.
Yeah, thank you. I think that something to research there. I didn't expect you to have exactly the answer, but I think you led me in a direction I can do some more research on myself.
Yeah, in fact, I worked in molecular biology at the University of Colorado, and we worked on yeast and looked at cell division cycle. That was the focus of the lab, one part of it. And if you didn't keep up with the research papers on a monthly basis, you would be behind. There's so much, and that was back in 30 years ago. It was that intense at that time, and I'm sure it's gotten even crazier now.
The second question is, from the beginning of the talk, what mechanism allows the transcription factor to find the particular gene that needs to be transcribed?
Again, I think that the transcription factor looks for the transcription factor binding site. And again, I think that might just be due to Brownian motion, that the cell is small and it's pretty squashed full of proteins and things. So I don't think there's anything that actually directs it to go there. And again, I'm not sure. That's the part that I worked on, but I just worked on the section of how do you find what the sequence is that is the transcription factor binding site. And I use some mechanisms to be able to try to find out what those sites were. They know they exist, but they don't know what they look like. They just know about the size of them. And again, this is from about 40 years ago, that there was they knew they were there. They didn't know how to find what they were. So I guess I can't answer that one either. But I would guess that it's Brownian motion, that the transcription factors are released because they're like, for the heat shock, the transcription factors would be transcribed, but in order to come over to the transcription factor binding site, I think would be through the Brownian motion. I don't think there's anything that carries that protein along to put it onto the DNA where it needs to go.
The next question is, how do these processes of forming proteins and formation of microtubules happen so fast? It seems they move too fast to be random collisions.
Again, I'm not sure. That's all what's the kinetics inside the cell? Well, the cell is pretty crowded. The picture I had showed you is actually not as crowded as it really is. So I'm not sure how those proteins get around and how they find what they're looking for, or if they're even looking for it, if it's done by chance. But the idea that how do these things find each other inside the cell? There might be something recent that I haven't looked up, but I don't know for myself.
I believe there was I'm not sure if it's by the Discovery Institute or not, but there was some real nice visual or a video of the microtubules and the kinesins moving along it. And as they step along, if they did have a collision, they could sidestep and kind of, work around the tubule and, bypass another one or keep from having a collision. It seemed like I had seen that on just recently.
Yeah, Discovery Institute has a very similar movie as the Harvard people do. That it's very similar to what the movie I was going to be showing you. The movie I was going to show you came out a long time ago. So this was, I think Discovery Institute came out with a more recent one that has similar pictures and similar things to it. Any other questions?
I had kind of another unanswerable question. We're always talking about the information within the DNA, and we're talking about all this production of, RNAs and mRNAs and all these different proteins running around. But is there, I was reading something about the reticulum being a place where maybe it's kind of like it collects the trash, the things that are not formed correctly and are, extra bases and amino acids and all that maybe congregate, the rough reticulum.
Yes, the endoplasmic reticulum. Yeah.
Yes. Am I close to asking a question there? Is what I'm getting at. Is there an excess of information there within the cell that, in other words, junk at that particular time, but can be reprocessed, reconstituted?
Yeah, that might be. And I'm not sure if it just gets sent out through the Golgi bodies and sent out because they're extra, or if they are floating around and can be used. I would guess that the cell would be pretty conservative and be able to reuse things that, if they made too many of the one amino acid or something, I would think that they would, the cell would be able to use that. But my other guess would be that it just gets secreted through the Golgi bodies that send things out through the cell wall.
Thank you. Mm-hmm.
We received another question here. Hold on, it just scrolled up on me. Is there any reasonable explanation showing how the complex structures in the cell formed by a materialistic process?
I can't think of any way that a ribosome can be made up of ribosomal proteins that have to be made by ribosomes. Even that one thing is pretty, I don't see how that could be by probability at all. And when you look at how all of the different molecular machines and the DNA and how it's made and transcribed and all the different systems, I just can't see how that could have happened by chance. I think it's pretty obvious that it's a designer, because so much of, when you just think of, for example, the mitochondria, how that takes the sunshine and puts it into the glucose in the plant, which is designed, and then I would think if it was by chance, somehow you could use that energy in the glucose molecule, but you can't. And you have to transform that into a different system using the mitochondria. And so it just seems way too complicated. And if you do the probability on even any one of those systems, I don't think the age of the universe is old enough for that to have happened by chance.
I hadn't seen anything that came close to that in any literature I've read. I just wondered if there was some literature was out there that people think, oh, for some reason they got that process worked out.
What's interesting is that they are still using kind of stories, almost like just so stories on how the species evolved from one to the next, or how this happened and that happened. For example, the flagella, they say, oh, well, there's a secretory 3 system that kind of looks like that. And so all it had to do is extend the flagella out there and move this secretory system over there and add these proteins. And then voila, you've got a flagella. But, they're almost just like descriptive, but there's nothing analytical. Or my background, besides the molecular biology, is statistics and probability modeling of using math to model biological systems. And I haven't seen anything on anybody saying this happened by chance and this is how it happened. And the chance is reasonable. I just haven't seen that.
I mean, that's what I strongly suspect. I come from an engineering background. I've yet to be able to design a system by putting parts in a box and shaking them until they get something useful out. But,
yeah, my husband uses the idea of a mousetrap. If you take the bar and the plank and the little hammer and all of the pieces of a mousetrap, and you put it in a box and shake it up, how long do you have to shake it before all of them happen to fall in the right areas in the right way? And that's like 6 parts. And we're talking about, thousands or tens of thousands of parts for one cell.
yeah,
So it's just unreasonable to think it could happen by chance.
Just getting a sequence of proteins. So if you know the probability of sequence of proteins, 20 different proteins, by the time you get a protein of 100 units, you're already kind of talking past the number of one part and the number of atoms in the universe.
Yep. It multiplies pretty fast.
We have another question here. Are all ribosomes the same, and can they read any mRNA?
I think so, unless there's something recent, but I think that ribosomes just are RNA readers, and just they're and they have that be able to put the amino acids on. So I think they are all the same. I don't think there's any difference between them. But again, that's a guess.
I think a lot of the new means out there of generating new proteins and stuff like that is basically kind of using ribosomes from different bacteria to make what you want. So I think that's a pretty common.
From different types of cells.
A name for it, but I can't remember.
Yeah, the single cell, like bacteria, the lower organisms. What I'm showing you is in a higher organism, the mammals, I think, or something. So the bacteria might have a simpler system than the cell that I showed you. There's prokaryotes, which are simpler systems. They don't have a nucleus. The DNA is just a circular piece of DNA, and they don't have chromosomes. So the prokaryotes of bacteria are just simpler things. I'm talking about more the eukaryotes, which is what I showed you to begin with, the algae, the yeast. Those are higher organisms. Humans are the highest. And those cells are more complicated.
Betsy, I wanted to ask you about something. So we look at all the complexity in the cell, and we want to go through this process of figuring out what's the most important, what's the basic, most critical thing. And, then you might say, okay, well, maybe something like the tubules aren't, the microtubules maybe aren't the most essential. Maybe, something could survive with a little less structure, or, maybe the junk would find its way out. You got to try to cut something, right? But then you look at it, and the microtubules are used for the separation of the chromosomes, and it just seems like everything's essential in one way or another.
Well, the example I have was that one protein on that nuclear pore didn't function properly, and that killed the whole cell because that one protein on the nuclear pore wasn't working. And so the cell couldn't divide properly. And so it essentially died. So every piece is important, every protein, every gene.
Okay, another question here. Mitochondria and chloroplasts do the opposite. So how can they have a common evolutionary pathway?
And why would they have to have a common evolutionary pathway? That's another reason where you'd think that, how could that happen by chance? One goes one way and then the other one undoes that and does makes it so that higher organisms can get that energy. The plants did it from the sun, but the animals get it from the plants and pretty much undo what the plants did to get it and put it into another energy formation that higher organisms can use. Again, how could that have happened by chance? And regardless of where you look, if you just look at the higher end of just looking at the cell and saying, oh my, that looks pretty complicated. And then what I showed you and talked about, well, yeah, that looks more complicated. And if you take a couple years in molecular biology, it's even more complicated. And this is all information from numbers of years ago when I was working yet. I'm retired now, so I'm sure that's even more systems and control systems and feedback mechanisms that they've discovered since then even.
That's all the questions that I see.
Yeah, I have one other question, Betsy, since you've done the statistics as well. I'm always struck by, to me, what seems to be the inadequacy of the Brownian motion explanation, and probably there's just a ton of stuff I don't have any clue how it works. But if you're waiting for what's essentially a random event, I don't understand how very complex sequences that are repeatable if you have the same structure, stressor in an environment. And bacteria are responding the same way. I always get this explanation, well, there's not a transport mechanism, or there's not a sense mechanism, or there's not a regulatory mechanism. You're relying on Brownian motion. Well, it seems that Brownian motion's awful dadgum lucky. Right? Because things happen in the same sequence, in the same timing, given the same circumstances in say different cells or different bacteria, the same species and things like that. So I've always found that to be a thoroughly, from an engineering standpoint, unsatisfactory explanation. Yet when I go out and look and research in white papers and stuff, that's the only explanation I get. So is there things about Brownian motion that are just that can defeat this probability problem that things are going to show up at just the right time because of stochastic motion inside the cell?
The only thing I can think of is I was working in a physics lab temporarily. They were doing biophysics. And Tom Perkins in Boulder here. And he was doing a motor protein, working on a motor protein and talking about Brownian motion. And he said, in a cell under Brownian motion, being hit by a water molecule is like being hit by a bowling ball going 100 miles an hour. It's just very, very different kind of, there's no momentum. It's just a whole different way of physics, the whole different physics at that level, molecular level, than what we're used to here in where there's a lot of air and things aren't quite as crowded and tiny. So that you would think that I would agree with you, and I do agree with you, but you'd have to go down to the level of the physics at a molecular level to really look into how that would work.
Yeah.
I see we have one comment there on the Brownian motion, mentioning it's powerful because it's fast and collisions are frequent. And I guess I would add one more thing to that, that there's also diffusion gradients. So if something's being generated in one spot and it's being used at another spot, there's kind of a source and the sink. And so the Brownian motion tied in with entropy move things around. And so things get to where they need to be eventually. It's not very efficient, but entropy and diffusion gradients do help. Well, it sounds like that comment totally destroyed discussion. We're probably near the end, so I don't know if there's something else wants to say, but I do want to say at this point, thank you for all joining us. And we do plan to have more of these in the future. We don't have a fixed schedule at this point, but we are in the Colorado chapter discussing what we might do next, and there is interest in continuing. So keep watching and we'll be seeing what's happening. And in that time, Betsy, is there another comment?
Yeah, Betsy, can I make one more comment? I was looking something on the Discovery website. We're always having these discussions, how can it, how can these things happen by self-organization, by chance? And there was I found this, just one paragraph I'd like to read. It's talking about the basic DNA molecule, your sugar phosphate backbone with the bases attached and tied together with hydrogen bonds down the middle. Anyway, from the website, almost reading it, the difficulty using self-organization scenarios to explain the origin of biological information can be illustrated by examining the structure of the DNA molecule. And there are bonds, for example, between the sugar molecules and the phosphate molecules that form the twisting backbones of the DNA helix. There are bonds fixing these individual nucleotide bases to the sugar phosphate backbone at each side of the molecule. However, there's no chemical bonds and thus no forces of attraction between the bases that run along the spine of the helix. That is precisely along this axis of the molecule that the genetic instructions in DNA are encoded. So there's not the forces of a natural attraction, a chemical bond that makes all this work and all that. Anyway, that's something I had found once, and I'd like to have a discussion on it further and all that. But that seemed like that answers that question a lot more than just the unlikelihood of chance and all that. Thank you for letting me.
The other thing for that too, in regards to that, is when they were trying to decide what the structure of the DNA was, they kept thinking that the information was on the outside of the DNA molecule. And they went forever and ever trying to figure out how that phosphates would give information. And then they finally realized, when they finally figured out the instructions, the information is on the inside of the helix, and it has to be pulled apart to reveal the information to make the RNAs to send out to make the proteins, which they had no idea that's what it was. And that delayed the structure of the DNA forever, because common sense would tell you it would be simple and the information's on the outside because it'd be easy to get at. But it's not simple. The information's on the inside, and you have to have more machines to pull the DNA apart, and then other molecular motors that untangle the DNA when it gets super tangled downstream because you're pulling the DNA apart to transcribe the DNA into RNA. And so the more you look at it, just the more complicated it gets.
Thank you, Betsy.
Any other questions?
All right, at that point, I'll say once again, thank you for joining us. Keep watching the website and we'll advise you when we have another presentation offer.